Good afternoon, everyone. My name is Aishwarya and I'm GRF at CIBioD. So, welcome to our today's session on nanotechnology in healthcare. Before we start our session, we would like to play a short video on CIBioD. Thank you.
Thank you, Aishwarya, for sharing this video. Well, very good afternoon to all. I am Shivani Singh. So before we move further, I would like to introduce CIBioD with all the participants. So CIBioD is a healthcare innovation hub and startup incubator established by Indian Council of Medical Research at a premium medical research institute and hospital of global repute in India, that is Postgraduate Institute of Medical Education and Research in Chandigarh. ICMR CIBioD aims to promote innovation in medical devices, instruments, life sciences, healthcare technology, education, clinical research, and healthcare delivery by creating a conducive ecosystem with the involvement of stakeholders from research institutions, government and private healthcare sector, globally. So if I talk about the objectives of CIBioD, it is to promote innovation and biodesign of medical devices, instruments, and technologies, including life sciences and other branches of healthcare, by making healthcare affordable. It is to promote automated solutions and approaches for various common medical problems. It provides manpower training for researchers, clinicians, and faculty working in the area of medical sciences. And it's a consultancy center providing a solution to hospitals and business in the area of medical science. Now, I would like to invite Ms. Kanupriya to please introduce our worthy speaker for today's session. Over to you, Kanupriya.
Thank you, ma'am, for such a wonderful introduction of CIBioD Center. Good afternoon, everyone. Before proceeding this session, I would like to briefly introduce our keynote speaker to the audience. Dr. Bharat Bajaj is currently serving as Assistant Professor at Center for Nanoscience and Technology, Panjab University. Sir has earned his Master's and PhD from South Korean University. He's a recipient of many prestigious awards, such as Young Scientist Award from SYST under DST, India. He's a winner of overseas programs from University of Science and Technology in Chicago, USA, as well as a postdoctoral fellowship from University in South Korea. Recently, Sir has been awarded and recognized as one of the prestigious UGC faculties among 50,000 faculty members at Delhi Technology University. His main research focuses on synthesis, fabrication, and characterization of electrospun carbon nanopipes. We welcome you, sir.
Hello, am I audible?
Yes, sir, you're audible.
Thank you. Thank you, Dr. Kanupriya, for a nice introduction. I'm sharing the screen. Is the screen visible to all?
Yes, sir, it is visible.
Okay. So first of all, thank you so much, all of you, to be here for this presentation. And thank you, Dr. Kanupriya, for a nice introduction. Well, today I'm going to talk about nanotechnology in healthcare. And as you all know, that nanotechnology is the science of extremely small, and it holds enormous potential for healthcare, from delivering drugs more effectively to diagnosis diseases more rapidly and sensitively, and also delivery vaccines via aerosols and patches. Nanotechnology is the science of materials at molecular or subatomic level. There are many unique health benefits of nanoparticles, like molecular imaging using nanoparticle, which help to detect, quantify, and display molecular and cellular changes that happen in vitro and in vivo. Especially these fluorescent biological probes using organic dyes are used conventionally in biology because of their inert qualities and their ability to interact without loss of sensitivity in a variety of cellular reactions. So I will give you an overview of what I'm going to talk about. He already gave my introduction, I have did my master's and PhD from South Korea. I was a winner of Korea Research Fellowship. So whole of my master's and PhD is sponsored by Korean government. Before that, I did my BTech in chemical engineering, and I worked in National Institute of Pharmaceutical Education and Research, Mohali, as a production chemist for 2 years. And from working in NIPER actually inspires me to change my stream to nanotechnology, because most of the professors are working in nanobiotechnology and nanopharmaceuticals. Well, all of you are wondering that why, after living such a long time in Korea, I came back to India. I lived there for 10 years. Like, I went there in 2006 for doing my master's. I was working for, I got a job in Korea Atomic Energy as a scientist. I worked there for 2 years. Then I decided to do my PhD. It takes me 3.5 years to complete my PhD. And then I work in different research institutes of Korea as a scientist. And finally, before coming to India, I was teaching in Yonsei National University as an assistant professor. So what brought me back, purpose of me coming back is, like, after living 10 years, I realized that I don't belong to that place. Even though I have a very handsome salary, nice lab infrastructure, very comfortable life, but there is one thing which bothers me, is sense of belongingness. Even after all the facilities, I never feel that I belong to South Korea. I learned the language. I tried to mingle with them. I tried eating their food. Why I'm sharing this experience with you? Because most of you wanted to go abroad for your PhD. So you should understand what is the purpose, how things are, and what is the reality. So what happened with me is, like in India, we have Aadhaar card. In Korea, they have this national identification number, which they call alien card, alien registration card. So we, all foreigners, those are working in Korea, they call us aliens. So that was the biggest, drawback. I don't like this word alien. So this is one thing. And other thing is, I was so much connected with my home country that I made my mind that if after all this, I will go back to India. Actually, I have a dream project which I want to finish. That brought me back to India. I met Honorable Chief Minister of Madhya Pradesh that time. He was visiting Korea for some business meet, and I showed him my interest, that I am interested in opening a Carbon Health Valley project in India, like Silicon Valley in America. I want to start this Carbon Health Valley project where health and innovation meet. And I have discussed the same with Mr. N.K. Sharma, who is the sitting MP of Derabasi. And he promised me that, yes, if you come back, we will definitely start up and we will give you a place in New Chandigarh to start this kind of project. So that is my motivation to come back after several years. I'm still working on it. It's not still completed. I joined Panjab University as an assistant professor. Earlier, I was selected for UGC assistant professor, and I was deputed in Delhi Technological University. I taught there for 2 years. Now, today in this talk, I will talk about concepts of nanotechnology and nanomaterials, how nanomaterial properties varies from bulk material, nanomedicines. It's an emerging field of nanotechnology to healthcare, and nanomedicine impacts in drug delivery and targeting. And finally, one of my favorite topic, which is role of nanotechnology in combating coronavirus. You all are aware of this pandemic, and what nanotechnology role nanotechnology is playing in combating this. I will discuss in detail, and other applications of nanotechnology. So before I start, I want to tell you the difference between science and technology. When we talk about science, nanoscience, basically it's deal with the study of phenomena and manipulation of materials at atomic, molecular, and macromolecular scale, where property differs significantly from those which are at bulk scale. So you can say, when we talk about science, is basically the study and understanding of properties of nanomaterials. When we talk about technology, technology is defined as the design. When we talk about engineering part, like design, characterization, production, and application of structured devices and systems by controlling shape and size at nanomaterial scale. So this is the basic difference between nanoscience and nanotechnology. Both do the same thing. They control and manipulate of matter at nanometer dimension. So when I talk to a layman, what is nanotechnology? I give them an example. The size of nanoparticle is to a football as a football is the size of the earth. So if you imagine कि जो नैनो पार्टिकल का साइज फुटबॉल के कंपेयर में है और फुटबॉल का साइज जो अर्थ के कंपेयर में है, दैट इज हाउ वी रिलेट नैनो टेक्नोलॉजी. सो इफ यू ऑल जस्ट ट्राई टू विजुअलाइज इट, you can imagine how small we are talking about. It starts from 0.1 nanometer to 1000 micrometer. Our human hair is 100 micrometer. You call it small. Our pencil tip and ant is a 1 millimeter. A bee is a 10 millimeter. Human is like around 1 meter, 1 or 1.5 meter. When we talk about like in micro range, our bacteria and red blood cells, they are in between 1 micrometer to 100 micrometer. Now, when it comes to nano, antibodies, virus, transistor, molecules, DNA, they come in the range of 1 nanometer to 100 nanometer. And as all of you are aware that atom is 0.1 nanometer. In the bottom side, if you see smallest nanochips, MSV pores of 30 nanometer, PDMS microchannels, vials of MSV formulations, NPS chip, biomarkers, nanopores. These are the type of nanomaterial which are synthesized at that scale. And they are range of dimension spanned by NMS products. Now, Dr. Kanupriya, is my screen completely visible? Because I can see some symbols up on the like mute, stop video, security things. Is it coming on my window too?
Yes, sir. Your video is, your screen is okay. No issues with it.
Okay, thank you so much. Well, there is a close relationship between nature and nano. I always say this. Like, have you ever imagined that no one has reached the efficiency of photosynthesis in storing energy? Can you believe that? And no factory does water purification and water storage as efficiently as coconut tree and watermelon does. Their capacity, their size and capacity, if you imagine, you will be amazed. Like, wow. Now, human brain in principle store and process more information than today's supercomputers. Or you can say today's normal computers, we can say. It is unlikely for any movie camera to capture visuals more vividly than a human eye. Our human eye is the best camera. You can zoom it, you can zoom out, you know, you can scan, you can see multiple things in micro of seconds. So nature inspires us. If you want to work in the area of nanotechnology, observe nature and try to mimic it. That's how everyone does. And the last thing is this sensory receptors of dog are much more sensitive than sensors we have developed. How many of you have actually dogs or pets? You will be amazed to know that their sensories are very sharp and they are the best sensors so far. And now this is the size, this image, if you look at this image, this is the size of human hair, which is about 100,000 micrometer smaller than a nanofiber, wider than a nanofiber. The background image is a nanofibril, and this other image is a human hair. So can you imagine how small is nanofiber? So these are the few examples. I'm giving you these examples so that you all can, because I heard all of you are from different disciplines, so you can imagine and visualize how small nano material is if compared with the bulk material. Now, here comes a question. Can we see nanomaterials? Rima, I would appreciate if I can, audience can reply.
Okay, sir.
Can anyone guess? Can we see them? I just mentioned to the size, but can we see them? Anyone?
No, sir.
No, sir.
No, sir.
So we can't see, right?
Yes, sir.
Okay. I think we can see them. I think no, it's actually we can see them. Nanomaterial can be defined as the material with at least one of its dimension in the range of nanomaterial. Thus, the material need not to be so small that it cannot be seen. It can be a large surface or long wire whose thickness in the scale of nanometers. So based on these, nanometers are classified like 0 dimension, 1 dimension, 2 dimension, and 3 dimension. Now, if you look at 0 dimension to 3 dimension, nanomaterial could be defined as the material with at least, as I mentioned, one of its dimension in the range of nanometer. So materials that are nanoscale in 1 dimensions, they are layers such as thin films or surface coatings. And all of you can feel and see the thin films and surface coatings. Am I right?
Yes.
Yes,
So we can see, right? We can feel, we can touch. And materials that are nanoscale in 2 dimension include nanowires and nanotubes. We cannot see them. I can say we cannot see 2 dimensions. And materials that are nanoscale in 3 dimensions that are called particles, for example, precipitates, colloids, and quantum dots, and small particles of semiconductor materials. Now, when you talk about, this is my area of expertise. I worked on electrospinning nanofibers, so I brought this slide in the beginning. These are the application of nanofibers, which we are doing in our lab. We are working on energy devices, solar cells, water filtration membranes. We are actually working on smart filter membranes, which can clean water as well as do other purpose also. Drug delivery, tissue engineering, sensor. And one of my favorite topic, which I'm presently working is, I'm making a membrane which can actually clean the air, especially this NOx and SOx gases. And I'm also working on removing virus from the atmosphere using some smart membranes. So this is the present research working on. We have already submitted the patent of this work, and hopefully we will do it. So today, I will talk about nanotechnology approach to nanomedicine. You can see that all of these, wait. So as I said, nanomedicine is the emerging field of nanotechnology to human health. And there is approach to nanomedicine. For example, pharmaceutical and therapeutics, molecular imaging and diagnostic tools, which covers nano biosensors, tissue engineering, stem cell research. And
कुछ भी करेक्ट लिखते जोर से बजाते हैं
अगर ऐसे की रहना तब थोड़ा टूट भी सकता है ऐसे के शायद थोड़ा सा लीडर सूट करे
I can hear some voices. Can you mute them?
इस कवर का फायदा है कुछ
Rima, I can hear some voices. Can you mute?
Please wait. Yes, sir, it's done. You can start the session.
Okay. So nanomedicine is based on molecular knowledge of the human body. It involves molecular tools for the diagnosis and treatment of different diseases. Since nanotechnology began as futuristic endeavor, it started by applying mainly nanomechanical concepts of the body. The size domains of components involved with nanotechnology are similar to that of biological structure. That's why nanotechnology is an important tool for healthcare sector. Because of this similarity in scale and certain functional properties, nanotechnology is a natural progression of many areas of health-related research, such as synthetic and hybrid nanostructures that can sense and repair biological lesions and damages just like biological structures. For example, white blood cells and this wound healing molecules. Now, there is a rapid increase in the nanotechnology application to medicine in order to prevent and treat diseases in human body. When we talk, there are two kinds of drug delivery system. One is conventional, other is controlled. Right? Conventional system, you all are aware. So, conventional drug delivery techniques have some common limitations in systematic biodistribution, including toxicity to non-target tissues, difficulty in maintaining drug concentration. Moreover, literature states that about 60% of all drugs are poorly soluble. So, when drugs are not soluble, so what happens? It correlates poor bioavailability. So it will not be absorbed from our gastrointestinal tract to the bloodstream and never reach to the site of action. That is the biggest drawback of conventional techniques of drug delivery. Now, when we talk about controlled drug delivery, it seems like, can you see this beep beep? It's going inside. So we call them nanobots. What actually they do? Nanotechnology based delivery systems, they could mitigate these problems and additionally provide tissue or organ specific targeting with therapeutic action. Nanomaterial, the gift of nanotechnology, act as drug delivery, and you all can also say drug targeting systems. Why? Due to this smallness of nanomaterials, they are not recognized by the human body. They migrate through cell membranes beneath a critical size and are able to pass, hence develop the nanoscale fairies. We also call them nanorobots or nanobots, which transport high potential pharmaceutics precisely to their destination. Now, we'll talk about smart drugs. These nanoparticles are actually solid colloidal particles consisting of macromolecular substance that vary in size from 10 nanometer to 1000 nanometer. The drug of interest is dissolved, entrapped, adsorbed, attached, or you can say encapsulated into the matrix of nanoparticle. So depending on the method, how we prepare nanoparticles, nanospheres, or you can say nanocapsules, we got different properties and different release method of nanoparticles. It has been recognized that CNTs are the key building blocks for nanoscale materials, and their further application to medicine could be inevitable. And we found that even they can be highly used for the treatment of HIV virus, human deficiency virus. As mentioned earlier, there are drug delivery vehicles. So magnetic nanoparticles, which include iron oxide particles, can be attracted and guided an external magnetic field for drug and gene delivery, and further they act as a potential tool for diagnosis. In this regard, the magnetic iron oxide nanoparticle coated with polyethylenamine. Why we are coating it with polyethylenamine? To make it biocompatible and for the uniformity of the, so that nanoparticle don't agglomerate when they go inside the body, they will remain separate. And also they act as a functionalizing agent to anchor different biomolecule moieties like virus, genes, bacteria, others. So these nanoparticles serve as a potential gene carrier with high transfection efficacy for cancer gene therapy. These nanoparticles were tested as gene vectors with in vitro transfection models, tumor necrosis factor related, and induces apoptosis. So the magnetic nanoparticle were used as a gene carrier to transfect this trail gene into MCF7 gel. Our molecule, our drug to be released. Now, if you can see, there are different kind of system like thermotherapeutic process, which release drugs that are encapsulated on heating. Like the drug when reached in the body, when you give them heat, they release the drug. And the kind of nanoparticle we use here is a gold nanoshell and gold nanoparticles. Ideal wavelength of light is between infrared range of 800 nanometer. And when we talk about magnetic nanoparticles, drugs are bound to magnetic nanoparticles. They carry drug to malignant side with magnetic field. If you look at this bottom image, there is a barrier of tissue. In the bottom, there is a magnet, and this is a bloodstream. And magnetic nanoparticle, they are carrying the drug through the blood vessel. So what happened when they carry drug to the malignant side with magnetic field? They release the drug by enzymatic activity. Now I will show you a video how actually it happened. What is the effect of magnetic field to the specific site? This is the magnet. We pull all the carriers. Now there is an external magnet at the bottom outside the body. So specific molecules which are sensitive to the cancer site, they stick on the cell, and unbind particles move away with the help of external molecules. Thus, this method is the highly sensitive method because there is a translocation of biomolecules to the specific site. Now, there is an interesting area which is called molecular imaging and diagnostic tools. So, as you all are aware that nanotechnology offers a new imaging technology that provides high quality images, which is not possible with current devices. For example, different nanoparticles such as magnetic nanoparticle, quantum dot, and gold nanoparticles are being used as a contrasting agent. These nanoparticles are optimal diagnostic tools since they eliminate most of the vulnerabilities of the conventional imaging agents. Metallic nanoparticle, they possess immense potential as X-ray contrast imaging agents, owing to their potent X-ray absorption and also because of the low toxicity profile observed over short duration in animals. Recently, gold nanoparticles have gained significant attention owing to the potential biocompatibility, relatively low short-term toxicity, and high absorption coefficient and physical density compared with iodine. Therefore, there is a significant demand for the synthesis of these type of nanoparticles. Quantum dots are semiconductor particles of few nanometer in size, which have excellent optical and electronic properties. If you see carbon nanotubes, gold nanoparticles, quantum dots, nanowire, they are widely used for imaging and diagnostic tools. So similarly, if you see gold nanoparticle nanodots in the images, antibodies attached to the nanoparticle antibodies bind to cancer cell, color reflect when light hits the particle, and shape and size also affect the color. Same happens when we use carbon nanotubes. Antibodies attached to carbon nanotubes, current changes, and we measure the current, and it can be applied to the cancer cell and viruses. Quantum dots are semiconductor particles a few nanometer in size, having optical and electronic properties. And when these quantum dots are illuminated by UV light, an electron in the quantum dot can be excited to a state of higher energy. So thus, this light emission, which we call actually photoluminescence, as illustrated in this figure on the right, shows the color, and we can identify different phases. Now, what is MRI? Magnetic resonant imaging is a test that uses a magnetic field and pulses of radio wave energy to make a picture of organs and structure inside the body. Because our body is mainly consists of water molecules, which contain protons, magnetic resonance imaging is a non-invasive diagnostic tool that applies magnetic field to the heterogeneous composition of water in the organisms. So what happened? The water protons of different cells show different relaxivity rates and get translated into contrasting images. Nanoparticles with a magnetic core are attached to a cancer antibody that attract cancer cell. The nanoparticles are also linked with a dye that is highly visible in MRI. These are some common nanoparticles which are used in molecular imaging, like solid lipid nanoparticles, liposomes, micelles, nanotubes, metallic nanoparticles, quantum dots, dendrimers, polymeric nanoparticles, and iodinated nanoparticles. Now, what is a biosensor? This is one of also biosensor application which use nanotechnology, and we call it nanobiosensor as well. So biosensor is a self-contained integrated device that is capable of providing specific quantitative, or you can say semi-quantitative analytical information using a biological recognition element. So biosensor actually employ biological molecules such as antibodies, enzymes, carbohydrates, and nucleic acids to identify or follow the course of any biological phenomena of interest. Interactions such as hydrogen bonding and charge-charge transfer between the ligand and receptor molecules, coupled with readout techniques such as colorimetry. When we talk about colorimetry, if there is a change in color, we can recognize. There is a other way of detecting is fluorescence, and Gmr Sensors, maybe you all heard about it. They read biomagnetic signals. They are used for sensing specific biochemical events and have a great application in food processing, environmental monitoring. You talk about clinical diagnostic, like measuring blood glucose. It's very common nowadays, right? And different nanomaterials like gold nanoparticle, magnetic nanoparticle, quantum dots, they are widely used because of their property and flexibility in changing the imaging contrast, as they are most favorable materials. But in comparison with gold nanoparticle conjugating probe, there are some smart material which are coming, which can act better. And carbon quantum dots, CQDs, are one of the best to up to the time best imaging agent. So examples of biosensors are cholesterol-based biosensors, antigen, antibody sensors, small molecules and ions in living things, DNA hybridization, nano and microarrays, and optical absorption. Now, nanotechnology in tissue engineering. Tissue engineering combines biology, medicine, engineering, and material science to develop a tissue that restores, maintain, or enhance tissue function. The application of nanotechnology in tissue engineering is discussed in terms of three effects: biomaterial, scaffolding, biomolecular manipulation, and cellular engineering. Now, when you go into a deep in a layman language, tissue engineering is a methodology and nanotechnology is a tool to create these nanofiber based scaffolds. So how we create them? There are two common techniques. One is the self-assembly, when nanomolecules, when molecules self-assemble them to create a new substance. And other one is electrospinning. In our lab, we are specialized in electrospinning of nanofibers and tissue engineering. We create artificial membranes which can be replicated as a human body part. So we are working on this biomaterial. Now, if I talk about present situation, COVID is actually spreading like anything. So how can we control COVID virus? As you're all aware, can I take a break for 2 minutes? Well, as you are all aware, that human coronavirus is also known as respiratory pathogen in layman language. It is associated with a range of respiratory outcome. That's why it is very dangerous. What it causes? Coronavirus causes an infection in your nose, sinuses, or upper throat. Most of COVID virus are not dangerous, but most coronavirus, when they first replicate in epithelial cell of respiratory, they are actually very difficult to handle. Coronaviruses are enveloped virus with a positive sense single stranded RNA genome with a nano nucleocapsid of helical symmetry. So you all know genomic size of COVID virus ranges from approximately 26 to 32 kilobases. That is the largest RNA virus. There are 4 genes of structural proteins like spike, envelope, membrane, and nucleocapsid. But now, using nanotechnology based treatment, we can actually detect COVID-19 infection and also treat them. So in this slide, if you can see how nanotechnology is helping in detection of COVID-19. The strategies for the development of novel nanotherapeutic materials for improving the efficacy of COVID-19 treatment, this includes fabrication of polymer nanoparticle with rapid and high mucus penetration feature, thus development of biodegradable, non-toxic and stable nanoparticle that could be used in lung with minimum pulmonary toxicity during treatment. Here I'm talking about testing or visibility. Recently, one of the group in US, what they have made is naked eye detection of COVID-19. What they do, they collect the sample through nostrils, and there is a then they put this COVID virus in a vial, and after then they add gold nanoparticles to this COVID virus, and after 10 minutes, they see that if the gold particles aggregate and settle down at the bottom, so this is the case of a positive, then this person is a COVID positive. And if it doesn't settle down, the person is a negative. So this is the very easy technique of naked eye detection of COVID-19 virus using the nanotechnology. Now, role of nanotechnology in vaccine development. Coronavirus enveloped virus with a positive sense single strand RNA genome and with a nucleocapsid of helical symmetry. It actually, how to say, basically a new generation of vaccine, you can say, called nano vaccines have been developed using nanoparticles to deliver antigens into the human body. It has been established that nanoparticles can target both adaptive T cells or B cells and initiate macrophages, monocytes, and neutrophils immune system at the cellular level. So if you look at this image, the nanoparticle can deliver and act as an intrinsic adjuvant with tunable physical loading of antigens. The COVID-19 mRNA-based vaccines are encapsulated in a positively charged lipid nanoparticles, enabling resistance to RNAs mediated, degraded, and stable self-assembling particle that can be injected via virus routes. There are a lot of future strategies for COVID-19, but before that, in our lab also, we have developed this antiviral coating, which can actually used on various materials, like which can used with water, wall paints, home appliances, fabric, PPE kits, and can kill virus in 30 seconds. The tests are under trial. We are waiting for approval. Our product is patented, and soon you will see this product in the market. We are also working on the rapid diagnostic kit in collaboration with PGI. And we made lab on a chip. People are working on lab on a chip, but now we are making lab on a chit. Chit means paper. We made a chit which is just a 20 paisa and can immediately detect coronavirus. And this work is also under trial. And soon we have already submitted a patent, and soon you will see this product. So see, this is a wonder of nanotechnology, that you can make things cheaper as well as efficient. So these are some of the work we have done in our lab as an application. Now, this is our research, which I would like to show with you. We have a flexible cholesterol biosensor, which is published on the cover page of Journal of Material Chemistry in 2016. We get this cholesterol tested on paper, and this is a paper-based nano generator. It's a nano generator. It can produce around 25 volt of energy, 1.9 to 2 volt of energy if you just bend it. But if you put a lot of pressure, it can even charge batteries. Another function is this: we are working on gas adsorption, separation of gas, or capturing of NOx and toxic gases. And also we make aligned and coiled fibers for tilt application. It's very difficult to, if you look at this image, last image, it's very difficult to manipulate fibers at this level. This is of 50 nanometer, means 1 billionth of your hair. And we assembled in a way that they are highly uniform in nature. And we also try to make coil-like structure of these nanofibers. We are actually trying to replicate DNA, or we are making nano springs. So this thing looks simple to you, but when you look into the dimension, it is of 20 nanometers, and the coil size is uniform. So manipulation at nanoscale is a difficult thing, but when you start doing it, it do wonders. Application we choose is for thin film transistors. This is our achievement, which I have earlier shown for cholesterol sensors. These are flexible nano generators. I have already showed you the details. We are also working on nano hybrids, which can actually kill the micropollutant in water. One of our paper got published on the cover page of New Journal of Chemistry recently. These are the nanofibers which I talked earlier. If you see, there are particles inside. If you can see the last image, you can see small particles are uniformly dispersed on the surface. They are around few nanometers. We are also working on super fibers. Basically, normal fiber can withstand approximately 150 kg of weight, and super fibers like carbon fiber, they can withstand 700 kg, but we are working on future fiber, which can actually withstand 1500 kg of weight. There are a lot of applications of carbon nanofibers. Our lab is dedicatedly working on applications of carbon nanofibers. There's a huge demand, but supply is very less. In Asia, North America and Europe, there are few companies, those are making nanofibers, carbon-based nanofibers. Toray, Japan is the biggest company. Those are contributing around 33% of carbon nanofiber manufacturing. These are some gobbler structures of nanoparticles on the surface of nanofiber. They can be used for many applications, such as gas absorption, biosensing, and other applications. We are also trying to make chameleon-like color-changing membrane. I told you earlier, nature inspires me a lot. I see things and I try to mimic it. I was very much wondered like how this chameleon changed the skin. So we are trying to replicate the skin, and when we play with our small line semiconducting chips, we got similar results. On the right side, we have tiny ridges of semiconductors, which can change when you flex, when you pull them out. The idea comes out because how this chameleon color change is a structural color. When they tightening their purple green screen, the color changes to red yellow, and when they relax, it changes to green color. So we try to mimic the same phenomena. And how we made it? We made color change membrane by stamping small lines into a flexible silicon membrane. Same phenomena is flexible silicon with a tiny ridges, and silicon bars or ribbons are very thin. You can say 1 by 1000 of the diameter of a human hair. And by stretching or squeezing the surface, space between tiny ridges, there is a change in color. Other is the turkey-like. If you have noticed, turkey skin tissues have collagens. They can change their color. So we also try to mimic this for making eye color analyzer, like for smartphones, use phage litmus, and we will soon publish this work. This is another color changing membrane based on bastard hogberry. It's a fruit which actually changes color because of this multi layers. So what we did, we mimicked the roll structure of fiber and a bilayer, and when we stretch it, we found that there is a change in color because of elasticity of a material. These are shape memory polymers. It regains its shapes after stretching, and we can produce electricity. These are some of the examples of what we did earlier. Now, this topic is also clotronic. This work has been done in Korea. I was working on clotronic, means electronics in clothes. You all are wondering how this fabric do wonders. So we were actually making stretchable conducting fibers, energy harvesting device using a zip, automatic temperature controller, and a dress play. Dress play is basically a display on a dress. You don't need a smartwatch when your sleeve can work as a smartwatch. So these are currently work happening in a lab. Fabric plus conductor plus stretch. I will not show that slide in detail, but this is one of the video which we have recorded in our lab, in which we coated nanofiber, which we made nanofiber through electrospinning and then simultaneously coating it. This work was done in Korea Research Institute of Chemical Technology, in which we make a very simple technology. If you see, fibers are coming out of this syringe. There is a bending. When we apply strong voltage, high voltage, around 20 kilowatt, these are other basins. if you see, the fibers are very small. They are coming as a jet because of bending instably due to electricity applied to the polymer. Now you can see this polymer is, the fiber is going in aqueous silver solution for plating. And then we have a reducing agent. There's a reduction of silver ions. And finally, washing of silver ions. It is very small fiber. You can't see by naked eyes. We use special cameras to capture this. So zip energy harvesting I already discussed with you. I guess my time is almost up. So I will, yes. These are some of the examples of auto temperature controller, how we made this smart fabric. And I will finish this presentation here. Thank you, all of you, for listening me.
Yeah, thank you so much, sir, for sharing your valuable time with us. The overall session is very interesting and informative.
Thank you.
So, thank you so much, sir.
Thank you. Are there any questions?
Students, any questions? I think, sir, there is no questions.
That's great. It's always good if there are no questions. Anyways, we are also looking for PhD students and master students. Yes, please.
Sir, actually, I can't understand about quantum dots. Can you please explain that?
What you don't understand about quantum dots?
Sir, you have told that quantum dots are semiconductor particles and few nanometer in size. About optical and electronic properties, you have talked about a state of higher energy in that. I can't understand that concept.
Okay. I will share. Maybe I have a slide which. Have you heard about quantum confinement effect?
No, sir. I'm a medical student. Zoology, botany का पता है, इसका ज़्यादा knowledge नहीं है.
Okay.
Yes, we are sharing.
I have this one, yes. So basically what happens in quantum confinement, in nanocrystals, the electronic energy levels are not continuous as in the bulk material, but they are discrete. Discrete means they are finite density of state. Because of the confinement of the electronic, wave function to the physical dimension of the particle, is limited. This phenomena is called quantum confinement, and therefore the nanocrystals are जो quantum nanocrystals होते हैं, they are called as quantum dots. इसमें क्या होता है आपका कि कंडक्शन बैंड और वैलेंस बैंड के बीच में जो डिस्टेंस है वो साइज डिक्रीज करेगा जैसे जैसे डिस्टेंस इंक्रीज करेगा. If you want to have more information, just Google it. It's a very common phenomena.
Okay, sir, I can get it. Thank you, sir.
Pleasure.
Hello. Good afternoon, sir.
Good afternoon.
Sir, my name is Shiva. I have a question that I belong to a computer science background, and I want to know that how a computer science student can contribute to a nanotechnology. Is there any scope? What could be research areas?
Very, very nice question, actually. One of our MTech student is a computer science student, and previously also we have computer science background students. I always inspire them. This is the era of multidisciplinary research. Software companies are hiring mechanical engineers. So can you imagine what they do? When they are mechanical engineer, they are doing software engineer. In computer engineering, if you are opting nanotechnology, human and machine interaction, is one of the very important topic in this present era. Many companies, especially in South Korea, Samsung, LG, there are a lot of projects in which they are working on robotics, human and machine interaction, IoT based things. So when you talk about smallest things, you see, I will give you an example. Can you see the size of a memory, like a memory drive? Earlier it was like 1 GB, 2 GB, right?
Yes.
Now you can get in like 64 GB, even 1 terabyte memory drives are there. How could it be possible? कैसे हुआ वो?
Reducing the size
Yes.
by semiconductors.
Yes, by reducing the size, but by generating the softwares. If you don't understand the nanotechnology, if you don't know how to manipulate things at that scale, you can't do anything. So being a computer engineer, if you want to pursue your master's in nanotechnology, you can opt for machine and man. There are specific courses also in nanotechnology you can opt for.
Okay. Thank you.
Pleasure. Anyone else?
I think, sir, there's no more queries.
Okay. So thank you so much, Rima. So can we wind up?
Thank you so much, sir. Students, I have an announcement that our next lecture will be on next Saturday, and it will be on chatbots. And all the details regarding this session will be shared via broadcast group and via mail. Okay. Thank you and have a nice day.
Thank you. Thank you so much.
Thank you.